Welcome to this video. We're going to be looking at our PowerPoint on key knowledge points 8 and 9. So we'll be focusing on DNA manipulation tools. Our study design points are as follows: the use of enzymes to manipulate DNA, including polymerase to synthesize DNA, ligase to join DNA, and endonucleases to cut DNA. Then we'll also be looking at the function of CRISPR Cas9 in bacteria and the application of this function in editing an organism's genome. So when we talk about DNA manipulation, let's be clear about a few different things here. Now, manipulating DNA used to be some far-off sci-fi fantasy, but it's actually pretty routine nowadays, and lots of labs do it, and it's the sort of thing that you might even get to do when you go to uni if you do a science course. So it's actually pretty standard nowadays. It's not that far-off futuristic science anymore. The reason we use it is it lets us understand the function of genes. It helps us to compare genetic information between species. We can even apply it outside of the lab and use it to solve crimes or paternity cases. We can use it in agriculture to produce transgenic plants and animals, helping improve the crop and animal breeds that are there. So those are just some ways that we can actually apply the manipulation of DNA and use it in real-world scenarios. There's 3 branches of DNA manipulation we're going to look at over this video and the next 2 videos and their corresponding PowerPoints. And those 3 areas are the tools, techniques, and applications involved in DNA manipulation. In this, we're going to be specifically focused on the tools that we use in DNA manipulation. And what we mean by tools are the different molecules that are used to copy, cut, and glue different bits of DNA together. It's important to note that if you're thinking of things like micropipettes or test tubes or like tweezers or scissors or those sorts of things, for our purposes, those are going to come under equipment, not tools. Tools are molecules in this scenario. So it's the different types of molecules that we're actually using. The 3 key ones we're going to look at are polymerases, endonucleases, and ligases. And you'll note there that endonucleases is one term for this type of molecule, but you might also see them referred to as restriction enzymes. And it is important that you're clear and able to use both of those terms and that you know that they are the same thing. So an endonuclease is also a restriction enzyme, and a restriction enzyme is also an endonuclease. The names are one's more common and one's more of a scientific naming. That's where sort of the difference between them comes in, but they're both quite commonly used. So let's talk about polymerases or polymerases. And that's kind of key. If you can break that down, you'll start to see what we're talking about here. We learnt back when we talked about enzymes that enzymes will put ASE on the end of their molecule name a lot of the time, not all the time, but quite frequently. So here we can see the ASE at the end of polymerase indicates it's an enzyme. So the question is, what comes before the ASE? Well, you'll see it's the word polymer. And that's because this molecule is involved in making polymers. So a polymerase is a polymerase. It is an enzyme that makes polymers. You've actually come across a polymerase before in the form of RNA polymerase. RNA polymerase is a polymerase where the polymer it's making is RNA. So that's what we add in the front there, is actually telling you what kind of polymer it makes. So it's a RNA polymer making enzyme is what RNA polymerase does. Another one is DNA polymerase. DNA polymerase is up there when you need to duplicate DNA before cell division, and its job is similar to RNA polymerase, make a new nucleic acid polymer. But instead of making RNA like RNA polymerase does, DNA polymerase makes DNA. So it's in the name there. There is another kind that you may come across, and that's called reverse transcriptase. And it's a polymerase, but it's got a bit of a funky name where you don't see the word polymerase in it. And that's because it does something a bit odd. RNA polymerase will take DNA, read it, and make RNA. Reverse transcriptase actually does the opposite. It takes RNA, reads it, and makes DNA. DNA polymerase, you might think would do that, but DNA polymerase will read DNA to make a new copy of the same DNA. So it doesn't actually convert between RNA and DNA. That's DNA polymerase. So to do that, you'd have reverse transcriptase. It's an enzyme that you'd find in some viruses, particularly retroviruses like HIV. They might have RNA in them, And when they infect a cell, they want to turn that into DNA to insert it into the cell's genome. And so what they do is they take the viral RNA and they use reverse transcriptase to make a DNA copy that they can then insert into the genome. So if you see reverse transcriptase, that's where that is from. It is still a polymerase, even though it's one that you may not come across as frequently as ones like DNA polymerase and RNA polymerase. Also, sometimes you'll find that polymerases get a different term in front, even though they might be a DNA polymerase or an RNA polymerase, just because they need to tell you something else about that specific polymerase. So we're going to talk specifically about DNA polymerase now. DNA polymerase, as I mentioned, its job is to read DNA to make more DNA. So what it's doing is it is reading the template DNA strand and making a complementary strand to that. And it just does this in a very similar way to how we've learned about RNA polymerase, which is involved in transcription. DNA polymerase goes along, it reads the template strand, it goes along in a 5 to 3 direction, reading that template strand, bringing in nucleotides, specifically deoxynucleotides, and it brings them in and attaches them to the template strand according to the normal base pairing rules, where A pairs with T and vice versa, and C with G and vice versa. DNA polymerase, as I said before, normally you see cells using it in replicating their DNA, particularly before they're going to go through cell division. So that's where we tend to see DNA polymerase in action. Now to work, DNA polymerase has to know where it's going to attach to this template strand. And to be able to do that, it needs something called a primer. And a primer is just a short segment of DNA that will attach to a specific region right before the DNA you want to copy. So if you have a specific bit of DNA you want to copy, if you pop in a primer that should sit right before that section, then that primer will go in, attach to the old DNA. The DNA polymerase will be able to recognize it, and it can jump on and start copying the bit of DNA that you want copied or you want replicated. Taq polymerase is a type of DNA polymerase that is found in bacteria that we would call thermophilic bacteria. Thermo as in heat, philic as in loving. So these are heat-loving bacteria. And scientists came across this polymerase in these bacteria and realized that they could use it to help amplify our own DNA through a reaction called PCR, which stands for polymerase chain reaction. So Taq polymerase is a type of DNA polymerase, but it's a very specific kind. That's why we don't call it just normal DNA polymerase. We're giving it Taq at the front to be specific about it. Because it comes from those thermophilic bacteria, it is totally able to withstand high temperatures, and it's really robust as an enzyme, which is great because it means we can put it in scenarios with high temperatures and it won't denature, and we can keep it working. So if you think back to when we talked about enzymes, because they're proteins, can denature under really high temperatures. And so Taq polymerase is a enzyme that its structure actually allows it to survive what we would consider high temperatures, which is why it's found in these thermophilic bacteria, because those bacteria live in environments with really high temperatures. So their enzymes need to have optimum action ranges that are really high temperature ranges. So that's a bit about Taq polymerase. So mentioned before, another type of a polymerase is reverse transcriptase. And as I was talking about earlier, this is a type of DNA polymerase where it will read RNA, single-stranded RNA, and it reads it and it creates DNA based off that RNA. And this is unlike DNA polymerase or RNA polymerase. They don't do this. This is going sort of backwards because we'd normally think of going DNA to RNA in transcription. This is the reverse of that. And so you end up creating a complementary DNA strand according to the template RNA strand that you have. And it works pretty much how you'd think it works. As the reverse transcriptase reads along the template RNA that you're looking at, the single-stranded RNA, it's going along 5 to 3 and it's bringing in nucleotides. Because it wants to make DNA, it's specifically bringing in deoxynucleotides. And again, it works according to base pairing rules, where if you have an A in the RNA, you'll have a T in the DNA. If you have a U in the RNA, you'll have an A in the DNA, and C and G follow their normal rules as well. Where it can be really useful is kind of reverse engineering things a little bit. If you remember when we make mRNA, so if you're in a cell and you've got your original DNA and that gets transcribed by RNA polymerase and you make your pre-mRNA, that will have to go through processing to become mature mRNA. Once you get it to the mature mRNA point, you've got a segment of RNA that codes for a protein and doesn't have any introns in it. What you can do if you have reverse transcriptase is you can basically get the reverse transcriptase to read this lovely freshly processed mRNA molecule to create a complementary DNA molecule to that, a cDNA molecule. And this new cDNA molecule will be a little bit different to the original DNA molecule because you'll have a DNA molecule that doesn't have any introns in it. So that could be useful down the track if you want to see what were the introns that got cut out. Basically, it's called reverse transcriptase as an enzyme. The A's is because it's an enzyme, and the reverse transcriptase means it's the reverse of transcription. Transcription is DNA to RNA. Reverse transcriptase goes RNA to DNA. So that is about reverse transcriptase. Let's talk about RNA polymerase. We've spent a bit of time previously discussing it because we've already gone over transcription, and RNA polymerase is the enzyme that facilitates transcription. So you should be relatively familiar with it already. It reads DNA, goes along, reads the DNA, it is reading the template strand, and as it's going along, it starts to make a complementary RNA strand to that template DNA strand. And it works by creating this chain going 5 to 3. It actually is reading the RNA 3 to 5, but because we're talking about two strands that are anti-parallel to each other, as the RNA polymerase moves along from the 3 to 5 direction along the DNA, the mRNA gets made 5 to 3. So going 3 to 5, it makes the thing 5 to 3. It's a little bit confusing, but you've got to remember the anti-parallel, so it just ends up like that. What you end up doing with the RNA polymerase is producing some sort of RNA. Now, we generally have talked about it making mRNA, as in messenger RNA, or pre-mRNA that then has to go off and be edited. It depends if we're talking, a prokaryote or eukaryote, exactly what comes off in terms of mRNA. But the RNA polymerase isn't just going to make messenger RNA. It is also going to make ribosomal RNA, that's your rRNA, and transfer RNA, the tRNA. So all those other RNA-based molecules that we have in a cell, ribosomes and the tRNA, they are also produced by RNA polymerase. And what will happen is you will have a segment of DNA somewhere that will contain the instructions for making this rRNA and tRNA. And so as the RNA polymerase reads those segments of DNA, instead of making mRNA, the RNA that comes off ends up either going to become a ribosome or to become transfer RNA. So there's not going to be as many areas in the DNA that do that as there are areas that will code for a protein and therefore produce mRNA. So just be aware that RNA polymerase can also make the other two types of RNA because its job is just to make RNA. Unlike DNA polymerase, you don't need a primer. We've learnt what we find with gene regulation is that there are promoter regions where the RNA polymerase will attach to, but DNA polymerase required a segment called a primer to indicate where the DNA polymerase needed to attach. RNA polymerase doesn't need anything like that to help it start synthesis. So it's a bit more independent like that. We use RNA polymerase when we want to look at transcription and amplifying RNA. Amplifying DNA or amplifying RNA simply means to make more of it, to increase the amount there is, to increase the volume. So that's what amplification means. So those were our 3 key polymerases. So you need to understand what a polymerase is and those 3 different kinds. All right. Now I want to talk about endonucleases or restriction enzymes. Both words mean the same thing. Like I said earlier, make sure you know both of those terms and know that they mean the same thing. These are enzymes that cut nucleic acids. Now, not all endonucleases / restriction enzymes will cut a nucleic acid in the exact same place. In fact, they look for different areas and they'll cut their own specific site. But what they do is they act like a giant pair of scissors, or rather a teeny weeny pair of scissors, because they're a molecule. And so they come along and they will cut up your DNA, for instance, or digest it into smaller fragments. And they cut the DNA at a specific sequence of bases. And that sequence is specific to that endonuclease. Different endonucleases cut at different sequences. So that's what that next point there says, that each enzyme recognizes a specific DNA sequence. We call that the restriction site, or sometimes the recognition site. And so once it finds that specific sequence that applies to it, it will cut there. What you find is the sequence you're looking for is also palindromic, so it is read the same forwards and backwards. Now, the restriction site isn't always going to be the same length. It actually varies based on the enzyme. So some enzymes will have a restriction site that's 4, some will be 6, or some will be 8 bases long. It varies exactly how long that restriction site is, just based on the enzyme. Because the sequences are shorter, any restriction enzymes that are only looking for a 4 base long restriction site is likely going to cut DNA more often than one that's trying to find a 6 base long cutting site. So you tend to find the smaller the restriction site, the more frequently that endonuclease will be able to cut up DNA. And you can see in the diagram down the bottom, just trying to highlight what we're talking about as a palindrome. So this goes, if you read the top row from 5 to 3, it goes GAATTC. Now you might be thinking, yeah, but if I read that backwards, I get CTTAAG. You actually need to look at the line below it. So if you read the top one, you go GATTC from 5 to 3 along the top. And then on the bottom, if you go backwards, which is actually still going 5 to 3 for the bottom row, you will go GAATTC. So it is actually palindromic in that sense. And that's what we're looking for, or what we mean when we say it's a palindrome. And it can be read the same forwards and backwards. We're talking about the top row, when you read it 5 to 3, will read whatever it does. And then when you read the second row in from 5 to 3, which is actually the other direction, you will also see that same sequence. So here are a couple of endonucleases. Now, it's worth noting that generally these are enzymes we find naturally in bacteria. And because of that, the way we end up naming them relates to the bacteria that they're actually found in. So you have HIND3 and 𝗃𝕃 or RV there. HIND3 is from Haemophilus influenzae, and because it was the third enzyme discovered from that bacteria, that's where the 3 comes from. So the HIND is, or HIN, D3 is just telling you what it's from and which enzyme number it was in order of discovery. It works by looking for the restriction site. You can see there where it goes AGCTT. So as soon as this enzyme comes across that sequence in DNA, it's going to make the cut you see in the diagram below. It actually separates the 2 A's and that are next to each other in the DNA. So it goes one cut there, and then it severs the connection between the A, G, and C and other T between the 2 sides. So it cuts through one side, one strand, and then it does a bigger cut to cut through between the 2 strands. So you end up with these weird sort of overlapping edges for this style of endonuclease. The other endonuclease we've got there, 𝗃𝕃, is from Escherichia coli, and it was the 5th enzyme, hence the V for the 5 at the end. Its restriction site is GAATTC, and it cuts a bit differently. It just cuts straight through. So where the T and the A are, it just cuts straight through from one side straight to the other and separates it with these 2 sort of blunt ends, we'd say, whereas the other one you've kind of got these overhanging ends because of that, it cuts it differently. This actually highlights the 2 different ways that endonucleases cut DNA. So these are 2 examples. They're good ones to be familiar with, and they're also handy to see the 2 different types of cuts that can get made. So when you use an endonuclease, like I was saying, they produce one of 2 types of ends, either a blunt end or a sticky end. Now, most endonucleases actually cut the DNA in such a way that they're producing sticky ends. The 𝕃𝕃𝕃𝕃 or 𝕃𝕃R5, it cuts it in such a way that you get a blunt end. That's where it just cuts straight through and there's no overhang. So all they're trying to do is cut through what we call the phosphodiester bonds in the sugar phosphate backbone. So all they're doing is chopping at the backbone. They're not trying to separate bonded base pairs. To make sticky ends, they actually not only do they have to cut through part of the backbone on each side, they also have to separate the hydrogen bonds between the paired nucleotides. So that's why we end up with these different ends. So let's look a little bit more about how this cutting happens. When you actually use a endonuclease / restriction enzyme and you cut up the DNA, you cut it into fragments. And if the DNA is circular versus if it's linear, like a straight line, like our chromosomes, then you're going to get different numbers of fragments. So if the DNA is circular, like a plasmid is, for instance, then the number of fragments you can make is equal to the number of restriction sites you cut at. So because it's circular, if you make one cut, it just opens it up and you've got one big fragment. If you make two cuts, you'll open it up and then you'll cut that in half, leaving yourself with two fragments. If you make 3 cuts, you'll end up with 3 fragments, 4 cuts, 4 fragments. So if DNA is circular, the number of restriction sites that actually get cut at will be equal to the number of fragments that get made. If, however, your DNA is linear or a straight line, like our chromosomes are, then it's a bit different. When you cut it, if you've got a straight line, you're actually cutting basically a straight line or straight molecule into 2 sections. If you cut it again, you end up with another fragment. And so what you see, the pattern is however many sites you cut out for these straight molecules of DNA will be equal to the number of fragments, but you have to add 1 onto that. So if you cut it once, it's 1 plus 2, so you'll end up with 2 fragments. If you cut it twice, it's 2 plus 1, so you end up with 3 fragments. If you cut it 3 times, it's 3 plus 1, so you'll end up with 4 fragments. So just be aware that there is a difference in the number of fragments you get per cut, essentially, depending on where you start off, whether it's circular or linear DNA to begin with. One thing to note, that your DNA that we're talking about, if it's double-stranded, each fragment is also going to be double-stranded. So you don't suddenly take your DNA that's double-stranded, cut it in, in half into 2 fragments or something, and then also turn it into single-stranded DNA. No, if you start with double-stranded DNA, you're just cutting really at the backbone to separate your pieces, and you're still left with double-stranded DNA. So just be aware of that. With the fragments you get left with, one of the things you often end up having to work out is how big are they? And the way we measure fragment size has to do with counting the number of paired up bases that are there. And we call these base pairs, or BP. Now, a base pair is just the 2 complementary nucleotides that are joined together across the 2 strands of DNA. So you can see there, I've shown with the green numbers how you would count these. So the first T and A, that is 1 pair. Then you go G and C, that's your second. AT, 3, CG, 4, all the way up, in this instance, to 22. So that fragment will be 22 base pairs long. Now that can seem pretty straightforward, but when you start to talk about fragments that have been cut with an endonuclease, occasionally this can get a little bit more tricky. So there's a slight difference between how you count those base pairs, dependent on whether or not you have a blunt end versus a sticky end. Now, thing to remember that you're measuring it in base pairs. If you have a base and it is not paired, it doesn't get counted. If you have blunt ends, this is super easy because they should all still be paired. So you can see with our blunt end example, where it gets cut at that green line, you end up with 1 fragment, that's 9 base pairs, and the second fragment, that's 13 base pairs. And every single one of those bases is in a pair, and so it gets counted. On the other hand, if you have it cut and it produces sticky ends, and you can see where the green line's going to cut to make that sticky end, you end up with something a little bit different. See, fragment 1 is still 9 base pairs, but fragment 2, it should say there, becomes 11 base pairs. And that's because we have these 2 bases on each fragment that are actually unpaired. So you can see on fragment 1, it ends with an unpaired A and T, and then on fragment 2, it starts with an unpaired T and A. And because they're not paired, they don't get counted. Okay, so make sure when you are counting out how big a fragment is, you are only counting paired bases. Something else you may come across as we are discussing endonucleases / restriction enzymes is something called a restriction map. And all a restriction map is it says here, if you have this specific DNA sequence, these are all the points that this specific endonuclease will cut at. And so you have restriction maps for different endonucleases based on whatever DNA sequence you are talking about. So this is really valuable because what it can let you do is work out, based on previous experimentation that has generated this, how many fragments and the size of the fragments you would expect a specific endonuclease to cut this specific DNA sequence into. The other thing is if you are adding 2 different endonucleases together, or even more than that, you can take those endonucleases restriction maps for your specific DNA sequence and you can combine them to work out where you expect all the cuts to be made, and therefore all the different fragments. Because if I cut with 𝕃𝕃𝗀𝗏1, in this case, not RV, it's RI, if I cut the DNA with that, and then I also cut it with HindIII, I'm going to get, presumably, all the cutting sites that the 𝕃𝗀𝑅I will cut at, plus the ones that HindIII will cut at. So I would expect if 𝕃𝗀𝑅I is cutting at 5 different places and HindIII is cutting at 7, then I'm going to expect there's probably about 12 places that the actual experiment will have cuts made at. And then I can combine them to work out how big those are. And so those numbers down the bottom, the 1 would be first base pair, and then say for 𝕃𝗀𝑅I, where the first cut is at 21228, that would be the base pair of that cut that's being made, so 21,226. Whereas for HindIII, its first cut would come at 23,130. So what I could work out here is that I'm going to get a cut at 21,226, so I'll end up with a fragment that's about that big. And then I'm going to end up with another cut, actually pretty soon after, less than 1,000 bases later, at 23,130. And so if I want to know how big the fragment is that's made between those 2 cuts, between 21,226 and 23,130, I just need to find the difference between those 2 numbers, and that'll tell me how big that fragment's going to be. If I was only cutting with 𝕃𝗀𝑅I, I wouldn't bother with this, but if I'm cutting with both, I have to make sure I'm looking at both values. If I was only cutting with 𝕃𝗀𝑅I, I could see that my first fragment is going to be from 1 to 21,226, and then my second fragment would be from 21,226 to 26,104, and so the size of my second fragment is between those next 2 numbers. So that's some way you can actually use the information in these, is to, before actually doing an experiment, work out the size of the fragments you think you'll get and what you expect to get. And that way you go into your experiment going, okay, if I'm expecting this, what did I actually get? And if are they the same or are they different? And if they're different, why are they different? So these DNA restriction enzyme maps are really, really useful for that sort of work. All right, we're going to take a bit of time now to have a look at 𝕃𝗀𝑅1 in a bit of action and how it works. And this brings us to exam question 1. Let's have a read of the question. It says the diagram below is a map of a bacterial plasmid showing ORI, the origin of DNA replication, and selected restriction endonuclease sites. One plasmid was mixed with the restriction enzymes 𝕃𝕚𝕚𝕃𝕚𝕚, PAM-HI, and 𝐻IND-CII. Which of the following shows the number of restriction sites that have been cut and the resulting number of DNA fragments produced? So you can see in the table there, you've got A is 3 restriction sites and 3 fragments. B is 3 restriction sites and 4 fragments. C is 4 restriction sites and 4 fragments. And D is 4 restriction sites and 5 fragments. So you will need to pause the video and have a bit of a think about this question and work out which one is the correct option. To see the answer explained, you can go and find the video on the answer to this question linked in the description box below. So you can pause the video now as you have a think about that. So let's talk about our third type of molecular tool in DNA manipulation, and that is ligases. Ligases, or DNA ligase, their job is to act like glue, where your polymerases act a little bit like photocopying machines and your endonucleases act like scissors. Ligases act like glue sticks, basically. And their job is to join DNA sequences together. They're essentially what we would say is they ligate these sequences. Now, if you've got blunt ends, this is going to be relatively easy. There's no overlap between them, so you should just be able to join the 2 blunt ends together. However, sticky ends provide a little bit of an issue, and sticky ends, remember, tend to be the more common end type that endonucleases make. So with sticky ends, the only way you can get them to be stuck together by DNA ligase is if they are compatible. So if this is happening in nature, you presume that this would happen, but when this is done artificially in a lab, what it means is that the 2 bits of DNA you're trying to stick together will need to have been cut with the same endonuclease. So basically, they need to have complementary overhanging sequences. So you can see in our example there that we've got 2 sticky ends being brought together, and they're able to do that because both of them have the same sticky end, essentially, even though they're from 2 different origins of DNA. They've come from 2 different sources. So you can overlap them, but if you just overlap them, nothing really happens. You need to have DNA ligase come along as an enzyme to help facilitate the joining of those 2 ends and stick everything together and seal up the gaps. Now, like I said, what's really important to understand is in the lab, if you are going to try and manipulate DNA and you're going to cut it with an endonuclease, and then you're going to try and stick 2 different bits of DNA together with a ligase, you need to make sure that those 2 bits of DNA you're trying to stick together were cut with the same endonuclease. The bits of DNA can come from different sources, but they should have been cut with the same endonuclease so that they end up with complementary sticky ends. Alright, so basically we use ligases to help recombine different DNA sequences. And if you wanted to insert DNA from one organism into another, this is the enzyme that's going to actually stick it all in place. And that's what ligases do. Now this brings us to exam question 2. Let's check how well your memory is going. DNA ligase does what? Does it A, join 2 fragments of DNA together by forming a phosphodiester bonds between the 2 fragments? B, acts as molecular scissors, cutting DNA molecules at specific nucleotide sequences. C, separates 2 DNA strands during transcription so that a copy can be made. Or D, is an enzyme involved in protein synthesis. So you can pause the video and have a think, and then you can go and check the answer video linked in the description box below to see an explanation of how to answer this question. So feel free to pause the video now. So we've talked about the tools that we use to manipulate DNA, our different molecules that get involved in copying or cutting or sticking together DNA. But there are different processes that we also will then use. So once we've used our tools, there are certain techniques that we'll also go on to. And these are processes that we might use them to amplify or to separate segments of DNA. Now remember, amplify means to increase the amount of, to increase the volume of. So that's what we're using the word amplified to mean here. The two types of processes that we're particularly interested in are PCR, or polymerase chain reaction, and gel electrophoresis. And you can see a couple little diagrams there. But we're actually going to go over them in much more detail in our next PowerPoint when we look at DNA analysis techniques. So this is just a little bit of an intro and an introduction to them as techniques. Same can be said for the applications, which we'll look in with greater detail at in our last PowerPoint. But when we talk about having tools, and which are the molecules we use to do things, the techniques, which are the processes we use to help work with DNA further, we want to think about why. What are the actual applications that we're going to use this manipulation of DNA in? And some of the key types of scenarios are in what we would call bacterial transformation. This is where we change up the DNA in the bacteria so that it does something else, and we transform what that bacteria is able to do. Also, in the manipulation of organisms to become genetically modified organisms or transgenic organisms, those two types of things are very similar, but we'll look at the difference between them in a later PowerPoint, particularly looking at GMOs and TGOs in agriculture. And also DNA profiling, which is a technique that police or detectives may use in hunting down a criminal, or even you use DNA profiling in paternity testing and that sort of thing. So those are some actual real-world applications where we can take those tools and techniques and use them to answer some other question. So that is the applications we're looking at, and we're actually going to specifically look at, with bacterial transformation, how insulin is made nowadays versus how it was originally made, and the whole role of bacterial transformation in that. Now, this brings me to a relatively recent tool that we didn't talk about earlier. We talked about polymerases and endonucleases and ligases, but I want to introduce something called CRISPR to you, or more formally CRISPR Cas9. Now, it was first identified as a bacterial immune sort of system back in 1993, so it was known about for a while. It continued to be researched in that time, and in about 2012, scientists were actually able to go, hey, we think we can take this thing that's part of bacteria and it's basically part of their immune system, and we can take it and manipulate it and use it to edit genes in other organisms, potentially even in humans. So we tend to think of this as relatively recent because it wasn't until 2012 that it sort of came into the public consciousness, that people actually became aware of this and its potential. But it was actually discovered back in 1993. Like I said, it's actually part of bacteria, and it's part of their immune system and how they defend against viruses, particularly viruses we would call bacteriophages. Bacteriophages are viruses that can attack and infect bacteria. So bacteria obviously want to defend against these viruses to protect themselves, and that's what CRISPR Cas9 do. So it's naturally occurring in bacteria, and it's how bacteria can essentially edit their own genomes to create a long-term memory of viral DNA and that sort of thing. So if the bacteria gets attacked by a virus, what will happen in the bacteria is they use the CRISPR Cas9 system to go and cut up some of that viral DNA, and then they take that snippet of DNA and they use it to create something called a CRISPR array, and they insert that into their own genetic material as sort of a long-term memory. And so basically these CRISPR arrays allow the bacteria a long-term memory. We have our own immune system that does its own thing in terms of remembering what you've been exposed to, and it's different to this, but it's the same idea that they want to remember and be able to, for next time, recognize that virus really quickly. And it's not necessarily that virus, it could also be really closely related viruses to the one they're exposed to. So what happens if the virus attacks again, the bacteria, because they've got this memory in their genome of the first attack, they're able to produce RNA segments from this CRISPR array, and they can use those RNA segments to launch an attack against the viral DNA that the virus is trying to insert into the bacteria. So what they end up doing is they use the Cas9, which is an enzyme, they use that to cut that viral DNA apart that's being inserted as part of the secondary attack. And by doing that, they actually end up disabling the virus. So that's how they can protect themselves. They get exposed once. They use certain enzymes to take a sample of the first lot of viral DNA and they put it into a CRISPR array in their own DNA. Then when they're exposed again, they can take a copy of that CRISPR array, create mRNA segments that copies of the CRISPR array, and they use that RNA segment along with Cas9 or a similar enzyme to then go and attack the secondary exposure with the DNA the virus is trying to insert into it, and they can go and disrupt that and disable the virus that way. So this is essentially what we're talking about. You can see there in number 1, the bacteria gets attacked by a virus. Number 2, enzymes in the bacteria locate something called the PAM, and we'll talk about that a little bit later, and they cut out the protospacer section of the viral DNA that is specific to that virus. So there's a few big words that are going to come up here. PAM and protospacer we can look at a bit later on. But they're looking for a specific region, and then they cut out a specific section. And that DNA that they cut out is specific to that virus that's affected them. Then in step 3, the bacteria is going to take that viral DNA they just took away from the bacteria, and they're going to insert it into their own DNA. And it's basically acting like a quick reference for future viral attacks. Now, in step 4, let's say that the virus has attacked again, or a similar virus has attacked this bacteria, days later. What happens is that section of viral DNA from the first attack gets transcribed into something called guide RNA or gRNA. Then the bacteria takes the gRNA and it attaches it to the Cas9 protein. And the gRNA kind of acts like a heat-seeking missile for matching DNA. And so the gRNA is able to guide the Cas9 protein to this new invading virus. And so the Cas9 protein can seek out those similar viruses because they'll have DNA that matches the gRNA that the Cas9 protein has. And so once the Cas9 protein is able to find a matching virus thanks to the gRNA, the Cas9 protein can then cut up the viral DNA that's being inserted as the virus is attacking and essentially inactivate that virus. So this is a step-by-step of how the CRISPR Cas9 system works in bacteria. This isn't how we use it in people or for editing other organisms. But what I want you to start seeing is we're talking about a system where you can provide an RNA segment to a protein so that protein go and seeks out the matching DNA. And that's the concept that scientists have taken and want to try and apply to use to edit genomes of other organisms. And we will look at that in a little bit more detail as we go along. So I've been using the term CRISPR Cas9 without explaining what it actually stands for, because you'll have noticed that CRISPR is written in all capitals, so it's actually an acronym. And CRISPR stands for Clustered, Regularly Interspaced, Short Palindromic Repeats. That's a lot of words, but it's actually not as tricky as it might sound. So we talked earlier about palindromes for restriction sites, and a palindrome is just a sequence of anything. It could be a word, can be palindromic, and it just means it's read the same forwards as it is backwards. So one of my favorite palindromes is a man, a plan, a canal, Panama. So if you read that forwards and backwards, that statement, that you'll see it says the same phrase forwards as backwards. So a palindrome in DNA is just a sequence of nucleotides that's the same in both directions. So that's what the palindromic bit means. And short means they're not particularly long palindromes. And so clustered means they're grouped, they're clustered, and they're regularly interspaced. There's regular things spacing them. Short, they're not very long. Palindromic, so they're a palindrome, repeats. There's a repeat in there, something's being repeated. And you can see down the bottom, I've got a diagram sort of showing what a CRISPR looks like. It's this stretch of DNA, and it goes repeat spacer, repeat spacer, repeat spacer, repeat spacer, repeat. And the spaces are those unique viral sequences that they've gotten from their first attacks. So the repetition is followed by the spacer DNA. That's the DNA that's been inserted as, it's been inserted to space out the repeats. And it gets inserted once the bacteria has been exposed to like viral DNA and that sample's been taken. So the sample gets taken and it gets inserted as a spacer between some repeats. So that's what a CRISPR is. It's this stretch of DNA where we see these palindromic repeats. So palindromic sequences, they're not particularly long, they're repeating, but they're interspaced. And they're interspaced with this foreign DNA. And they're all clustered together and the interspacing is regular. And that's what CRISPR is standing for. Cas9 is the name of the protein. Specifically, it's an enzyme. And what it does is it's like an endonuclease in that it acts like a pair of scissors, like a pair of molecular scissors. And so what it is able to do is cut up strands of DNA. What does Cas9 stand for? It stands for CRISPR-associated protein 9. And you can see I've put in bold where the Cas part and the 9 come from. So that's all Cas9 means. Some other words that I mentioned before were PAM and Protospacer. PAM stands for Protospacer Adjacent Motif. So adjacent means next to. So if it's Protospacer adjacent, it's next to the Protospacer. A motif is a short sequence of DNA. So PAM is a short 2 to 6 nucleotide sequence in the viral DNA that's right next to what we call the protospacer. I'll talk about the protospacer in a moment, but basically in the initial attack, when you need to first get a copy of that viral DNA, the bacteria is going to send out its Cas1 and Cas2 proteins. We tend to focus on Cas9, but there are other kinds of Cas proteins, and Cas1 and 2 will go out in the initial attack and they'll go looking for this consistent section that's always there and it's always right next to the protospacer. So these proteins go looking for this PAM, this sequence of DNA that's always right next to the protospacer. And when they find it, they know, hey, I found the point where if I cut it here, I've got the protospacer right next to me. And it's the protospacer that they want. So if they can cut at the PAM, they know that they'll have cut and out the protospacer as well. What they do is they're actually really good because they don't cut in such a way that the PAM gets included. They cut it just before the PAM so that it's excluded and you don't end up with it in the protospacer. So, for example, in Streptococcus pyogenes, the specific PAM sequence is NGG. Now, the N just means it's some nucleotide. So N for nucleotide. So it could be G, C, A, or T. Doesn't matter. But the other two are GG. So what happens is Cas1 and Cas2 will go looking at the foreign DNA for any nucleotide. It could be, like I said, A, T, C, G, then followed by 2 consecutive guanine. So they're looking for that little sequence. If they find, say, AGG, they go, that's my PAM. That is my short little sequence that I happen to know comes right next to the protospacer. So what they'll do is they will then cut it in such a way that, say, the AGG doesn't get included. And now they have their protospacer. Now, I've been using the word protospacer a whole heap without explaining it. You'll notice that it is proto and spacer. And we did just talk about spacers on the slide before. And the spacer is the bit of DNA that ends up inserted into the bacterial DNA. And that's the bit that down the track will get read when there's a secondary attack. Protospacer is just the form before it's the spacer. Often we use proto to mean sort of beginning or initial, like a protostar is the thing that comes before a proper star in the night sky. So a protospacer is the sequence of DNA that will turn into the spacer. It might just need a little bit of tidying up and editing before getting to that point. So that's all the protospacer is. It is that fragment of DNA that will eventually go into the bacterial DNA as the spacer. And it just needs a bit of processing beforehand. So we're going to take a little bit of time to have a watch of this video on CRISPR.
I keep hearing that CRISPR is going to revolutionize medicine, the way we fight disease, cure cancer, and maybe even create new humans. And I agree with that. But I haven't been able to find great videos out there that explain what CRISPR is. They tend to be too complex or too simple. So I thought I'd throw another video into the mix. I proudly present... Like most things in molecular biology, CRISPR was first identified in E. coli. And if we break apart the acronym, it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now that's a mouthful, but it does tell you the two main parts found in CRISPR. First of all, we have the repeats. These are going to be short segments of DNA, so 20 to 40 letters in length, and they're going to be palindromes. Remember, a palindrome is a sequence of letters that read the same left to right, like never, odd, or even. So we're going to have these letters that are palindrome. The reason why is that when you transcribe that DNA, you make RNA that forms these little hairpin turns. So we've got the repeats. Those are all identical, one after another, after another, after another, but they're interspaced. And so what's in the middle, we're going to have what's called spacer DNA. Now what's interesting about the spacer DNA is that it's not identical. Each segment of spacer DNA is going to be unique. And this puzzled scientists when they identified this back in the 80s and 90s. But in the 2000s, what they found is that that spacer DNA, that's the important DNA, matches up perfectly with viral, especially bacteriophage DNA. They also identified a number of genes associated with CRISPR. So these are the CRISPR-associated or Cas genes. Now those Cas genes will make Cas proteins. The Cas proteins in general are going to be helicases. Those are proteins that unwind DNA. And then nucleases, those that cut the DNA. And so the idea was perhaps this is an immune system for bacteria, a way they could fight their old nemesis, the bacteriophage. And that's exactly what's going on. So if we have a picture of E. coli, this would be the cell membrane, cell wall right here. This would be the genome of the bacteria. I'm highlighting the Cas and the CRISPR system. And so when the bacteriophage injects its DNA, what normally would happen if you don't have an immune system is this DNA would hijack the cell. It could become embedded in the genome, but more importantly, it would make a bunch of these bacteriophages and eventually kill the cell. But since it has this CRISPR system, what it's going to do is it's going to transcribe and translate proteins, so this Cas complex, and it's also going to transcribe that DNA to make what's called CRISPR RNA, and it'll fit right into this protein like this. What is this? It's a way to fight that viral DNA. It essentially breaks it apart, and so before the infection starts, the infection essentially has ended. Now you might say that's interesting, but what happens if it's injecting DNA where we don't have a spacer that matches? Well, the CRISPR-Cas system works there as well. It's going to create a different class of protein, a class 1 Cas protein, and what that'll do is it takes the DNA in, it breaks it apart, but more importantly, it takes that DNA and copies it into the CRISPR system. So what is CRISPR? It is spacer, repeat, but the spacers are essentially history of old infections so we won't be infected again. This is exactly the way your immune system works on a much larger level. You're making antibodies and then you have white blood cells that'll envelop that invader. But what scientists thought is if we could hijack this CRISPR system, we could perhaps use it, because this is a living cell here, to either inactivate genes or maybe even embed new genes. And so the search was on. And the one that you'll hear most about is the CRISPR Cas9 system. This was identified in the labs of Jennifer Doudna and Emmanuel Charpentier. And what she was working on was Streptococcus pyogenes and their Cas CRISPR system. And what's interesting about it is that they only had one Cas protein. We call that Cas9. Now it doesn't look like this, it looks like this, but if we look at its major structure, it has a nuclease. So it's got this section right here where it can cut DNA here, and it can cut DNA here as well. In S. pyogenes, they also are creating two long strips of RNA. We have the CRISPR RNA. The CRISPR RNA is going to fit into the Cas, but they also have what's called tracer RNA. So if we look at what that looks like in this bacteria, you've got the spacer segment. That's going to be the part that matches up with the corresponding viral DNA. You have this tracer RNA that essentially holds the CRISPR RNA in place, and then this whole thing together forms this complex where we can break DNA. But what the lab thought is, wouldn't it be cool if we could modify this whole system? Use the one Cas9 protein, but let's put our own sequence of DNA right here. And then if we could somehow connect these two together, we'd have a really simple system, and that's what they did. They created the tracer RNA, CRISPR RNA, 𝕃𝕚𝕤. And so what's a 𝕃𝕚𝕤? It's this ancient mythological beast that's a combination of all these different species. And so what they've done is created a new type of RNA, and they've got a system that's really simple. It's got two parts in it. You've got the Cas9 protein, and then you've got this 𝕃𝕚𝕤. And since we're making this simpler, let's just call this the guide RNA. These are the two parts of a CRISPR Cas9 system. This is going to be the CRISPR part. It's going to be the RNA that's got the information of where we want to cut. And then we've got the protein that's actually going to do the cutting. And this is what happens. And so if we've got a little bit of DNA, so this is the DNA that we want to cut, we create a guide RNA that's going to have a corresponding bit of RNA. What happens is the DNA will feed into it like that. Once it's in place, we're going to cut it right here, and we're going to cut it right here. And so we do this little snip, and now we have an inactivated gene. We've broken the gene. Now the cell will try to fix it. It'll do some insertions and deletions, creates mutations. But what we can do a lot of the time is we can inactivate that gene. That's what the bacteria are going to do. But since we've created it, we can cut the DNA wherever we want to cut the DNA. We essentially just have to know what is the sequence of DNA that we want to cut, put that into the guide RNA, and then we can cut it. Now let's say we want to make this more complex. Not only do we want to break a gene, but let's say we want to insert a new gene. Well, now the system's going to just have three parts. We've got the Cas9, we've got the guide RNA, and then we've got the host RNA that we want to put in. So as we break the DNA, the host DNA is going to be added, and then the DNA is going to fix it. So essentially we've added the gene to the cell. Now what's cool about the CRISPR Cas9 system is it does this in living cells, and it can cut the DNA in multiple different places. So how could we use this? Well, let's say somebody has cystic fibrosis. What we could do is use a system like this to fix the genes in that person. Or in the future, we could engineer a new embryo. You can kind of see where this is going. But more importantly, I hope you know what a CRISPR system is. In review, a CRISPR system is an immune system that was identified in bacteria and then modified in humans. And I hope that was helpful.
So we've spent all this time talking about CRISPR and what it does in bacteria, and it's important that you understand how CRISPR works in bacteria. But the question is, can we apply it to something else? And so remember, when we talked about editing DNA and our other molecular tools earlier, our endonucleases were the things that did the cutting. But you're restricted in where they cut based on that endonuclease's own restriction site. What happens if you want to cut somewhere and you don't have a endonuclease that will cut at that spot? That's a bit of a problem because suddenly you're limited with in terms of what you can do. So traditional endonucleases are really great in what they do, but they're also really limiting because they will only cut where they can cut. However, the Cas9 protein doesn't have the same restrictions. Remember, the Cas9 protein can cut anywhere the guide RNA tells it to. So what if you can make up your own guide RNA so that you can take the Cas9 protein exactly where you need it to go, so it cuts the DNA at the precise location you need it to be cut? So that's a way we could apply this process to editing the genomes of other organisms. So all we need to think about now is, can we make our own guide RNA? Because if we can make our own guide RNA and we can then attach it to a Cas9 protein, suddenly we've got our own little heat-seeking missile of a pair of molecular scissors. And that is exactly what scientists are working towards and have been doing. So let's have a look at how we can take this CRISPR-Cas9 system from bacteria and put it into a system where we need to use it to edit genes. Now, I've got a step-by-step here, so we're just going to sort of walk ourselves through it. The first thing you're going to need to do is create your synthetic guide RNA. Now, obviously, back behind the scenes, before you actually get to this point, you would have done a whole heap of research and you would have looked at the gene sequencing and worked out where it is you actually want to cut this DNA sequence. And from that information, you're able to make your synthetic guide RNA. Now, you make this obviously in a lab, and you make sure that it has a complementary spacer to the target DNA that you want to cut. Then you get your Cas9 enzyme, and you make sure it has the appropriate target PAM sequence. And that's because down the track, when that Cas9 encounters the DNA, it won't actually bind to it or be able to do anything unless it has the appropriate PAM sequence for right near where it needs to cut. So you make sure that your Cas9 enzyme has that appropriate PAM sequence so it can actually function later on. Then you take the Cas9 and you take the guide RNA you've made, you put them together and make a mix, and you bind them together to create this CRISPR Cas9 complex. Now we have our Cas9 has its guide RNA attached, and it's become its little heat-seeking missile for wherever that guide RNA is going to take it. The guide RNA and the Cas9 mixture will get injected into the specific cell. That might be a zygote, it could be whatever you're trying to target. The Cas9, because it has that PAM sequence that we made sure it had at the start, is able to find the DNA and find the bit where it wants to act on, and it's able to attach to it. And then it will also check that the guide RNA is matching with the DNA. Because those PAM sequences are so short, it might come across a PAM sequence that matches, but it doesn't match the guide RNA. So having the guide RNA there would mean, oh, whoops, this is the wrong spot. We don't actually want to cut here, and it stops it from doing that. So the Cas9 will go on until it finds another PAM sequence that matches. And this time when it finds that PAM sequence, hey, the guide RNA actually matches the DNA, it aligns with it, so this is actually our cutting site. So the PAM sequence is great for that initial seeking out of location in the DNA and binding to it, but the guide RNA is going to dictate whether or not it's the correct spot to actually cut the DNA. Now we're found our correct spot in the DNA, the Cas9, the enzyme itself, will actually cut the selected sequence of DNA at that point, wherever you have dictated it to cut by that guide RNA fragment. Now, as you cut this, it creates a blunt end in the DNA, and the cell will go and try and repair that because it doesn't want this broken DNA. Now, it's at this point where the DNA is trying to be repaired that we can get in and we can do a few things. So while that DNA is being repaired, you can actually get the cell to introduce new nucleotides into the DNA at its site. And these could be ones that you have specifically put in as sequences into the cell in the hope that it will ligate or be attached into that gap. So basically what you end up doing is you use the CRISPR Cas9 system with your made up guide RNA to find a section of DNA that you want to cut open. Once you cut that open, it's just a process of getting the new bit of DNA that you want to insert into that point. And this may take a few goes, or you might have to try it on a few different cells before it actually happens. There's no guarantee that the bit of DNA you insert as the new stuff to put in will actually end up being ligated into that gap that the CRISPR Cas9 makes. But you keep trying in the hope that it does, and you keep testing to see if it's worked. When you do this, there are three main results that can happen. You can disrupt, delete, correct, or insert. So that last one's kind of they're kind of similar thing. So disrupt means you have made a single cut, and a process that's called non-homologous end joining can result. This means that there's no template that the DNA will be repaired along. So when you cut when the CRISPR Cas9 system cuts the DNA, you're left with these two broken or separated ends. And generally, those ends get somewhat degraded and they're not following any pattern as they get reattached. So non-homologous end joining. These ends aren't following the normal homology or pattern that they should be. And what that means is you can actually lose base pairs. Sometimes you can get them added in, but overall, your sequence gets messed up. And generally this inactivates the gene. If you want to switch off and knock out a gene, then this would be something that you could use. Another thing that can happen is if instead of making one cut, you make two, you could deliberately cut out a segment of the DNA. You just need to make sure you're using two guide RNAs, so you're targeting two different points to make those two cuts. If you make those cuts, or cleavage in the sense this means you cut or to separate, after making those two cuts, again, non-homologous end joining, these two ends are not following a template, they just get pulled together, unites these different ends, and it means that you suddenly have removed this sequence of DNA from your original sequence. You've removed this smaller section. And so you've deleted it. This again can be another way to switch off, or rather to knock out a gene completely by removing it. So for those first two, disrupt and delete, you end up generally disrupting and removing some functionality or some gene from the system, from this original section of DNA. The last one, where you can correct or insert, is where you make the cut, but you insert a DNA template. And that DNA template, alongside with the CRISPR-Cas9 machinery, means that the cell isn't working blind to try and fix things once the cut's been made. So the CRISPR-Cas9 makes its cut, just like it does, but you now have a section of DNA that's going to help the cell repair that cut. And so that's what that DNA template is for. And that template may just be correcting a singular mutation in the DNA that you've cut. It doesn't need to be something big. Or you could actually be inserting a whole new section of DNA, a whole new gene could get put in. This process is called homology-directed repair. And this process allows us to either correct or completely insert new DNA and to fix things up. So we're going to watch a video to look at this all in a bit more detail.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene, or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics, or check out some of the other mind-blowing science being developed around the world and in the universe right now.
Sickle cell anemia is a great little case study in a sense of how we can use CRISPR Cas9. So we'll follow through our little diagram here. We've met Susan. She has sickle cell disease, and it's caused in her DNA by just one letter being mutated. So it's a singular base pair that's wrong. So you can see she has a DNA double helix with a sickle cell mutation. So what happens in the lab is the scientists are going to create a strand of guide RNA that matches that mutated sequence. They take that guide RNA and they mix it with Cas9, which is they're going to be the molecular scissors. Susan then gets an injection of this RNA and Cas9 mix, and in theory what we hope would happen and what should happen is that Cas9 will be attached to that guide RNA, and the guide RNA will be able to identify and direct the Cas9 to the mutated DNA sequence that's causing Susan's sickle cell disease. The Cas9 can then go in and cut out that affected sequence, and as long as the scientists have also given in that injection a healthy DNA sequence of the correct gene, then Susan's cells will be able to repair that cut the Cas9 made with the correct DNA sequence in there. In theory, if all goes smoothly, what we see is that Susan's DNA has been edited from an incorrect mutated version to the correct version, and she would no longer be suffering from sickle cell disease. So this isn't necessarily something that's happening, but this is something that in theory, this is how we could apply this process. To help you get to grips with it a bit more, I want you to take some time now to go and do the CRISPR Cas9 online activity here. In particular, work through the how it works segment. The how it's used bit is great for getting a feel of all the different places we can apply it, but it's also a whole range of videos for you to watch. So the how it works is the interactive activity part, and the how it's used is a bunch of videos that you're welcome to watch to see all the different potential applications for it. You can find the link for this activity in the description box below. So you can take a moment, pause the video, go and do this, and come back later and finish watching up the video. So hopefully you've now had a chance to go and do that CRISPR Cas9 activity. I want to take some time to look at some of the limitations and ethical issues around this type of technology. Now, CRISPR Cas9 is super powerful. It's way more powerful in theory than our traditional endonucleases, but there are limitations to it. You see, one of the problems that we have with CRISPR Cas9 is a similar problem that we have with traditional gene editing tools, and that is once you have cells that are mature, say in an adult human, they've got lots and lots of cells. If you want to try and manipulate the DNA in those cells, that's a lot of cells that you're trying to get to, and it's pretty difficult to get the CRISPR Cas9 material to all those mature cells in large numbers. You can use viral vectors, which is where a virus has been basically emptied out and all the problem stuff for that virus has been removed. Good stuff that we want to use has been put in, and we use the virus then as a transport or a vector to help deliver the material we've put into it. So viral vectors are pretty common, but they're not completely perfect, and so it's still really difficult to get this CRISPR Cas9 stuff to lots and lots of cells. Also, it's not completely 100% efficient. Just because you get a cell that takes in the CRISPR Cas9 material, it doesn't mean that that CRISPR Cas9 material is actually going to edit the genome of that cell. So that can be a limitation too. Not only is it not completely efficient, it is also not completely 100% accurate. And this is definitely something you want to be 100% accurate if you're going to use it on people. And generally speaking, it's highly accurate. But you can still get what are called off-target edits. This is where something is edited that was not the target. It's off the target somewhere else. And even though these may end up being rare, they could have really severe consequences, particularly when we're talking about a clinical application. So using this with people to treat a disease, for instance, like treating a genetic disease like sickle cell anemia. So there's a big question mark over it with that. If it's not completely accurate and it has the potential for these off-target edits, what issues could that actually cause? Could that do something to someone that kills them? And that's a pretty big question mark to have hanging over it. And of course, there are the ethical implications. Ethical implications at the core of it is often asking, is this right? Should this be happening? Is this right or is it wrong? And so gene editing in general raises a lot of ethical questions and concerns. And these can come from a number of different viewpoints in terms of, well, does it cause harm, or is it within my worldview and my religious beliefs, or is it something that contradicts them? And so people come with lots of different ethical concerns about things from a lot of different positions. And gene editing taps into quite a wide range of them. Now, with CRISPR, some of the issues there have to do with the fact that if you want to have CRISPR edit the genome for an individual, it would be best to do it in germline cells for future offspring. The problem with that is, if you are manipulating the genomes of gametes and subsequent offspring, they don't have any say in it. There's no informed consent. And so some people really dislike that. Or the editing of a zygote, of a recently fertilized egg, very, very early on in development. You could edit it then too. But that zygote never has a say in whether or not it wants that to happen. So you can see they've sort of broken some of the key areas of concern into 5 categories. The idea of safety and unknown consequences, of informed consent, justice and equity, the use of embryos in research, and also this idea of vanity editing. So safety and unknown consequences raises the issue of those off-target effects. If there's a potential for edits to happen in the wrong place, should we allow this technique to be used? Is it okay? Is it right to use this knowing that there could be very serious off-target effects? That's one concern. Is it right to use this technique if it could lead to accidental mosaicism? And this is where you end up with an organism whose cells, some contain one set of DNA and some contain a different set of DNA. So it's like they're two people. And as the third point there, is it right to use CRISPR given it has unknown health consequences? Now, I will say particularly on that last one, that you've also got the question of, is it right given all the benefits that we know that it could bring? So ethical issues are never perfectly black and white with a nice clear answer, and rather they tend to be questions that you have to engage with and consider all the views and values on. Informed consent deals with the idea that for a medical procedure to happen to an individual, as much and where they are able, they should be fully aware of what is happening and the potential consequences for it before they are able to consent to it. Now, I say so much as they are able. There are times, and this is true in medicine in general, where you have an individual who is unable to provide informed consent for various reasons. And that might just be they don't have the mental capability, and so generally they have someone who will provide that for them, who works on their behalf. As a kid, this happens as well. Your parents are the ones who provide the informed consent because as a kid you're not able to, and often as individuals get older, if they're still unable to for whatever reason, they will have someone who's a carer who does that for them. So if we're talking about editing someone's cells such that their germline cells and their gametes will be infected, what that means is their offspring will also be affected and never get to consent to that. And the other issue around informed consent is, given there are potential unknown consequences, how informed can your consent actually be in this issue? Is it informed enough? Thirdly, our third category is justice and equity. Doing something like this is not necessarily cheap. So, how do we make sure we don't create an entirely new class system of those who have the ability to have gene editing versus those who don't? This is very much the premise of the movie Gattaca, which you can go and watch. It's an oldish movie, but it deals with this idea of a society where a lot of people, before they're born, have favorable traits edited in so that they are the optimum that they can be, and it creates a class system in that society. And so, does CRISPR pose the potential for creating a new class system? Will the technology be available to everyone, or only those who can actually afford it? Because like I said, it's not cheap. So that's the issues with around justice and equity, or some of them. You also have the issue of, as we're doing this research, it often does involve using embryos. And a lot of people have real issues with this for moral and religious objections, because it is seen as the destruction of potential life. A curveball with that is often these embryos can come from IVF couples who no longer want them, and otherwise these embryos would be thrown out. So is it then maybe ethically better that we use them so that they do contribute to something and they're not completely being wasted? You have that. So you've got different views on this, and it's really interesting. And the last point here is a really interesting one. It ties back a lot into that idea of CRISPR providing the potential for a new class system. And that's the idea of vanity editing. I think a lot of people can see the benefit with CRISPR being used to help people who have genetic disorders and diseases that are really debilitating. But we don't tend to have a problem with that, because what we see is often somebody who, something is seriously affecting their life in a negative way, and we want to help them. And if we can do that through CRISPR by editing their DNA, we don't tend to see that as a bad thing. But I suppose, where do you draw the line? If someone feels that their eye color is negatively affecting their life, would you allow them to use CRISPR to modify their DNA to change that? Vanity editing is this idea of using something like CRISPR to make modifications that satisfy one's own vanity, not because they're actually curing a disease or anything like that, or treating something. They're just being used to enhance desirable traits. Instead of getting lip fillers, you manipulate your genome so that your lips are naturally plumper. That would be vanity editing. And is that right? Should we allow that to happen? So there's all these issues, and this is just sort of touching on the surface of it. But because there's so many issues around CRISPR and gene editing in an ethical sense, it's been a key reason why scientists globally have called for what's called a moratorium, a complete halt and stop on human germline editing until these ethical issues and societal implications are more fully understood and accounted for. And I've included an article there that you can go and read. It's from Nature. It's really interesting to have a look at where this moratorium came from and why they wanted it in place. So that's a whole range of ethical issues for you to think about when it comes to gene editing. And like I said, it's not even all the potential issues that there are. So this brings us to our third exam question. And it says, the CRISPR technique is a gene editing method. It involves a protein called Cas9 and a short piece of guide RNA, gRNA. The gRNA leads Cas9 to a gene in the DNA that scientists wish to edit. Cas9 is an endonuclease that acts in a similar way to a restriction enzyme. A. What action is Cas9 expected to perform? And then DNA ligase is required for the next stage of the editing process. B. What role would DNA ligase play? And our next bit, the CRISPR technique can be used to insert new genes or to delete sections of a gene. While some scientists have recognized the potential benefits of using the CRISPR technique to edit genes in the future, other scientists have expressed concern. C. You need to state one potential benefit some scientists may have recognized and one concern other scientists may have had. So you've got a spot there for the benefit and a spot there for the concern. So I want you to go pause the video, go away and think and answer these 3 questions. And then you can check your answer and see how to answer this question in the video in the description box below. So pause the video and go do that now. Alright, we're going to now watch a video talking about Cas9. It's a bit of a fun one. It's a song. So we're going to have a watch of that now.
CRISPR Cas9, bring me a gene, encoding for a specific protein. Make a few snips at this coded locus. You work so well inside a streptococcus. Cas9, I'm so alone without your scissors in my chromosome. Cut me up and do it clear. CRISPR Cas9, bring me a gene. CRISPR Cas9, keep me a gene, a viral sequence you've already seen, chopped into bits and stored as genomic, with clustered repeats that are palindromic. Cas9, bind with this code, use it to target infections of old, immunize like a vaccine. CRISPR Cas9, keep me a gene. CRISPR Cas9, cut me a gene with a precision that I've never seen. Unzip a strand and interrogate it. Seek out your sequence until you locate it. Cas9, lock into place and do your job as endonuclease. Chop just like a guillotine. CRISPR Cas9, cut me a gene. Snip, snap. CRISPR Cas9. CRISPR Cas9. CRISPR Cas9, bring me a gene by commandeering my repair routine. A strand to match your severed location for some homologous recombination. Cas9, cheap and precise, rewriting genomes from microbes to mice and soon the humble human being. CRISPR Cas9, bring me a gene. CRISPR Cas9, give us a gene, give us a miracle like that one Nazarene. Cause giving the lame their legs and the blind their sight is. If you put dystrophy and retinitis, with CRISPR Cas9, why did you fall? Outside our power and inside us all, that really good inside a scene. With this terrible, wonderful power unsettling arms, the door to an ethically metal is a congenital malady, better to fit to warrant genetics and better to modify man in the matter of getting a recipe, a mammoth or make a radical grammatical medical means to eradicate aging or cancer or make a fanatic or massive epidemic, no weapon to bury a single mosquito, no weapon malaria, send in a viral infection to bury a cure to the sense of an HIV carrier, freedom diseases were free to recover for nature and accident failed to discover and try to be other than all that we ever did. CRISPR Cas9. Cas9, bring us a gene.
Now this brings us to the very end of our video looking at DNA manipulation tools and CRISPR Cas9. If there was anything you didn't understand, you can always go back, either rewatch that section, or you can reach out to your teacher for further clarification. Otherwise, I'll see you in the next video. Bye.